Hello friends, welcome to my course on microbial biotechnology. We are in module number 9 where we are discussing food production involving microorganisms and their products. In today's lecture, we are going to discuss about the health benefits, risks and advances in food fermentation. The lecture is broadly divided into two sections. In the first section, we will discuss about health impacts and advances in fermentation, and in section 2, we will be discussing about the precision fermentation. So, let us straight away go to section number 1, which is further divided into health benefits and risks of fermented foods and modern biotechnological advances in fermentation. Let us have a small discussion on probiotics and gut health. Probiotics maintain gut balance by promoting beneficial bacteria and inhibiting harmful pathogens. So, they are very important for our health. Different probiotic strains offer distinct health benefits. So, the beneficial properties are actually strain specific. It helps in digestion. Fermented foods improve digestion, nutrient absorption and reduce bloating by restoring gut microbiota. It has also an immune function. Probiotics strengthen the gut barrier and modulate immune responses, supporting overall health and reducing infection risks. It can also help us in addressing chronic diseases. Probiotics may help prevent chronic diseases including metabolic and inflammatory disorders. Now, if we want to describe what are probiotics, they are live microorganisms, mainly bacteria, that offer health benefits when consumed in sufficient amounts. They are found in fermented foods and supplements, and we already know about the various characteristics and health benefits the probiotics offer us. From the nutritional point of view, fermentation helps us in increasing vitamin content, and also the antioxidant properties are enhanced. And then we have certain vitamins which is enhanced by the process of fermentation, and the bioavailability also increases, particularly for B12, B6 and folate, making them easier for the body to absorb, which is especially helpful for those with dietary restrictions. Certain fermented foods such as natto significantly increases vitamin K levels, particularly vitamin K2, which is vital for blood clotting, bone health and cardiovascular health. Fermentation also boosts the antioxidant properties of foods, which may help reduce the risk of chronic diseases by combating oxidative stress. The effect of vitamin content varies depending on the bacterial strains used in the fermentation process. So, all these properties are actually strain specific. So, let us look into the increased bioavailability of nutrients and the beneficial compounds. So, enhanced nutritional absorption is increased by fermentation, because it increases the bioavailability of nutrients by breaking down complex compounds, making vitamins and minerals easier to be absorbed. For example, lactic acid fermentation reduces phytic acid, enhancing mineral absorption, particularly iron. There is also production of beneficial compounds by fermentation. Compounds like vitamins and bioactive peptides. Lactic acid bacteria, for example, synthesizes B vitamins, boosting the nutritional value of fermented foods. So, however, there are certain risks associated with fermented foods in spite of the many benefits it offers to us. For example, there may be contamination, so which can lead to pathogen risks. Fermented foods can be contaminated by pathogens like Escherichia coli, Salmonella and Listeria if not properly prepared or stored, leading to foodborne illnesses. Contamination can occur during fermentation or through improper handling, and then it may lead to dysbiosis and health issues. Pathogenic contamination can disrupt the balance of beneficial bacteria, leading to dysbiosis and gastrointestinal problems. This imbalance undermines the positive effects of the probiotics. Then there are certain populations which can be actually vulnerable to these risks, particularly pregnant women, infants and individuals with weakened immune systems are especially vulnerable to infections from contaminated fermented foods, risking severe gastrointestinal illnesses. And proper fermentation techniques, strict hygiene, correct storage temperatures are crucial to prevent contamination. Regular monitoring for pathogens ensures food safety. Then there are also the risks from allergens and intolerances. For example, some people may be allergenic to dairy products. Fermented dairy products like yogurt and cheese contain casein and whey, which can trigger allergic reactions in sensitive individuals. Then certain grain-based foods which are fermented, such as sourdough, may contain gluten, which may pose risk for those with celiac disease or gluten intolerance. Then there are fermented soy products like miso and tempeh, which can cause allergic reactions such as skin, respiratory or digestive issues in vulnerable populations. Then certain fermented food may have high levels of histamine, which may cause headaches, rashes or digestive problems in those with histamine intolerance. Then there is the risk of cross contamination. Fermentation cross contamination can introduce allergens like gluten into otherwise safe products. Then there is a risk of excessive consumption of fermented food, which may lead to digestive issues like overconsumption of fermented foods can cause bloating, gas and diarrhea due to high probiotic and fiber levels. And certain fermented food has minor alcohol content. For example, kombucha contains small amounts of alcohol, posing risks for those sensitive to alcohol. And there is also the issue of religious belief or other health reasons due to which these kind of food may cause certain risks or problems. Then there is nutrient imbalance. Excess intake, especially of dairy based ferments, can lead to nutrient imbalances, including increased saturated fat and calories. Then there may be a risk of probiotic overdose. Excessive probiotics may overwhelm the gut, causing issues in immunocompromised individuals or disrupting gut balance. Then there is a risk of food spoilage, microbial contamination, particularly improper preparation or storage of fermented foods can allow harmful bacteria like Salmonella or E. coli to grow, causing foodborne illnesses. And there is a risk of mold growth due to improper storage of products like cheese can facilitate the growth of mold, which may produce harmful mycotoxins, unlike the intentional mold in the blue cheese, which has some commercial value. Then there may be off flavors and textures resulting out of the fermentation process or due to the contamination. Spoilage alters taste and texture. For example, yogurt can become too sour and sauerkraut may turn mushy if not properly fermented. Inadequate pH reduction during fermentation may allow spoilage organisms to thrive, such as in kimchi, lacking sufficient acidity. And then storage is very, very important. Inconsistent temperatures like overheating kombucha can cause uncontrolled fermentation and spoilage. Let us now discuss about some modern biotechnological advances in fermentation. Let us start with the genetic engineering, particularly the genetic improvement of yeast, where we undergo targeted modifications that allows precise changes in the yeast strains, such as deleting or overexpressing specific genes to improve performance and product quality of the fermented foods. Then there are certain cases where genes from other organisms are introduced into yeast to enhance dextrin and starch breakdown, increasing ethanol yield while retaining the flavor. And we also go for the modification of genes like ILV3 and ILV5 to reduce unwanted byproducts such as diacetyl, which causes off flavors in beer. Then we also want to improve the flocculation of the yeast in the final product. This is done by overexpression of flow genes, which enhances yeast sedimentation, improving beer clarity and production efficiency. However, despite the vast potential, the use of genetic engineering in brewing faces regulatory and other challenges like consumer acceptance. So, much research remains to be done in different fronts. Let us now discuss about the genetic improvement of bacteria, which particularly focuses on the bioconservation. We use antagonistic microbes or their substances, example bacteriostacins, for natural food preservation, replacing chemical additives. Then we also can build up bacteriophage resistance, genetic techniques, enhance phage defense capabilities, preventing fermentation losses. Then the engineered strains can produce customized exopolysaccharides, improving food texture, stability and yield, and optimized proteolytic activity enhances flavor and texture in dairy products, including producing bioactive peptides. We also have metabolic engineering approaches. We alter the metabolism to enhance desirable compounds, example diacetyl acetaldehyde, and remove unwanted ones using CRISPR and plasmid systems. And we also try to address challenges like stress by using genetic modifications. We improve the tolerance to heat, acid and oxygen, boosting fermentation performance, etc. Some of the examples of genetic improvement of bacteria include Lactococcus lactis, which is well characterized for different milk fermentation. Some of the focus is genetic engineering to enhance production and control. Then we in Lactobacillus species, which is a diverse species used in various fermentations, genome sequencing has aided in better exploitation for traditional and novel uses. Then we have Streptococcus thermophilus, which is used in yogurt and cheese production. Genomic research is improving strains for large scale processes. Then we have Leuconostoc species, essential in vegetable fermentation, where again genomic insights has enhanced flavor production in industrial applications. Then we have Pediococcus species used in meat and vegetable fermentation, plasmid analysis and genetic studies for strain improvement. And then there is another example of Onococcus oni, which is key for malolactic fermentation in wine. Genome sequencing has been undertaken to refine fermentation control and improve the wine quality. One of the important work that has been done is in the strain improvement of lactic acid bacteria, particularly lactic acid bacteria in dairy fermentation. Lab is known for their safe use in milk fermentation, and health promoting properties are key components of human gut microbiota and widely used as probiotics. Due to their safe and beneficial properties, lab are suitable candidates for genetic modification in food, industrial and pharmaceutical applications. So, here we have two approaches. One is without genetic modification and another is genetic modification. So, in the first case we use random mutagenesis, or we go for adaptive laboratory evolution, and also food grade plasmid transfer through conjugation. So, these can be done with the help of starter cultures, probiotics and research tools, and of course, these act as cell factories. Then in the GMO, genetically modified organism approach, we go for plasmid based homologous recombination and then recombinering, and use modern tools like CRISPR-Cas genome engineering. And this is an overview of the genomic modification tools and the applications in improving the properties of the dairy fermentation. So, what do we do in adaptive laboratory evolution? This is a technique for evolving strains with improved traits by long term culture under specific selection pressure. It is widely used to enhance strains, particularly lab strains, for fermented dairy products without being classified as genetically modified. And so, there is the advantage of not getting rejected by consumers who are little bit conservative about GMO foods. And beyond strain improvement, ALE aids in understanding gene function, regulation and bacterial population evolution. So, some of the applications of ALE is thermotolerant Lactococcus lactis mutant for reduced seeds ripening time. We have more cut down in the cost in terms of time and investment. Then we have the lactic acid bacillus rhamnosus GG mutants with enhanced freeze thaw viability, and acid tolerant lactic acid bacillus casei mutants which can survive lethal pH conditions. In rhamnotop mutagenesis, we introduce random genome mutations characterizing numerous variants and selecting mutants with desirable traits. This method, widely used in fermented dairy production, avoids classification as genetically modified microorganisms. Some of the examples are the use of UV, where for mutagenesis a Lactobacillus helveticus mutant was produced and found to have improved yogurt texture and reduced post acidification. Then we can go for mild mutagenesis with high throughput screening combined with microfluidic droplet technology. Lactococcus lactis variants were identified tripling riboflavin production during milk fermentation compared to wild type strains. And we can also go for heavy ion mutagenesis. In this case, generated Lactobacillus thermophilus mutants with high lactic acid production was obtained. So, briefly from the application point of view, precision fermentation supports the sustainable production of enzymes, lipids, vitamins and flavoring agents often more efficiently than traditional methods. Then another approach is food grade plasmid transfer through conjugation. Conjugation is a natural DNA transfer process where plasmids move from a donor to a recipient cell via cell to cell contact through a pilus. In lab, particularly Lactococcus starters, conjugated plasmids carry transfer genes, TRA genes, and encode traits like lactose metabolism, bacteriophage resistance and bacteriocin production. And when it comes to the applications in the dairy industry, some examples include the transfer of PMRC01 encoding the bacteriocin lacticin 3147 improved starter strains by preventing non-starter lab outgrowths during cheddar seeds ripening and inhibiting Listeria monocytogenes in cottage seeds. Then there is the stacking multiple bacteriocins in starter systems which enhances food safety. While most applications focus on Lactococcus conjugation, has shown potential for transferring traits to other lab species such as from Lactococcus lactis to Lacticacibacillus rhamnosus GG. Here in these plasmid based homologous recombination, we have the integrative suicide vectors which contain an antibiotic marker and two contiguous homologous regions HR1 and HR2 that allow integration into the bacterial chromosome having homologous regions. These vectors cannot replicate in the recipient and therefore must integrate to maintain antibiotic resistance. After transformation, a single crossover event occurs by the first homologous region. A second crossover then excises the plasmid, resulting in either a wild type or mutant genotype, depending on which homologous region is involved. Non-replicative vectors can efficiently modify the genome in this manner. However, it suffers from certain limitations like low efficiency, and it is also a very time consuming process, and often we end up with high false positive rates, and there are issues with foreign DNA in the food grade. Let us now discuss the next approach, which is recombinating. Recombinating developed for E. coli enables precise genome editing using fast derived recombination systems. It integrates oligonucleotides into genomes with single stranded DNA or double stranded DNA templates. Key systems include lambda red beta gam exo and RAC prophase derived RAC E/T, which facilitates DNA annealing and protect introduced DNA from degradation. Some of the applications with respect to lab is in Lactococcus lactis and Limosilolactobacillus reuteri, using RACT mediated ssDNA recombination, mutagenesis has been achieved with efficiencies up to 19%. And then in DS double stranded DNA recombinering, we have found that large deletions are possible up to 6 kb, and gene insertions demonstrated in LactiplantyBacillus plantarum and Lacticacibacillus casei has been reported. The next most interesting application is that of CRISPR-Cas9 based genome engineering in lab. The CRISPR-Cas system is basically an adaptive bacterial immunity mechanism, which has become a powerful tool for genome engineering. The Cas9 nucleus guided by RNA, as you can see in this picture, targets and cleaves DNA at specific sequences. These are basically flanked by the photospacer adjacent motif sequences. This result in the double strand break, as you can see over here, which are actually lethal and will kill the cell in the absence of a repair system. In many lab strains, the repair system required for DNA repair are absent or inactivated, making CRISPR-Cas9 system an effective counter selection tool as wild type cells are killed by Cas9 cleavage. However, in the presence of a repair system, the cell will survive. So, CRISPR-Cas9 assisted recombining requires two plasmids, one for the recombinase machinery and another for Cas9 and single guide RNA. The recombinering template can be single stranded DNA or double stranded DNA or plasmid based. The dual step procedure is more efficient than the single step method. This system has been successfully used in various bacteria, achieving high recombination efficiency and resulting in the edited mutant genome. Some of the applications of CRISPR-Cas9 in lab is the recombinering, where CRISPR-Cas9 assisted recombinering improves efficiency for single stranded DNA and double stranded DNA editing, achieving up to 100% success in Limosilolactobacillus reuteri and LactiplantyBacillus plantarum. Then also we have the cases of base editing, where catalytically impaired Cas9 variants coupled with cytidine deaminase allow CG to TA substitutions, which is used in Bifidobacterium lactobacillus lactis. Some of the applications in lab include CRISPR interference or CRISPR-I. These catalytically inactive Cas9 silences genes via transcriptional blocking. It has been demonstrated in Lactococcus lactis and LactiplantyBacillus plantarum. Here in this figure you can see the gene silencing, which is using catalytically inactive dCas9 together with guide RNA to cause a transcriptional blocking of the RNA polymerase, leading to the knockdown of the gene expression of the target gene. CRISPR-Cas9 has revolutionized genome editing in lab with precise modifications, but faces challenges like plasmid instability and varying efficiencies. Niki's variants reduce lethality and boost recombination, while endogenous CRISPR-Cas systems, both type 1 and type 2, simplify editing and lower cytotoxicity through requiring thorough characterization. These advancements enhance genome engineering for research and industrial applications. So, in this table we have summarized the comparison of lab genomic modification tools from the points of view of advantages and disadvantages. So, some of the advantages of random mutagenesis: no recombinant DNA required, applicable to all lab strains and species. This is not considered as GMO, but disadvantage is that untargeted and introduces unintended mutations randomly, requires high throughput screening limited by existing genetic potential. In the case of adaptive laboratory evolution, the advantages we have is It is not considered as a GMO. It also do not require recombinant DNA applicable to all lab. It can help us in achieving directed evolution, but disadvantage is that it is again like random mutagenesis, untargeted and co-selects undesired changes, contamination risks with long evolution times and limited by the genetic potential. In the case of food grade plasmid transfer, we can get targeted modification. It is a natural method, also considered not as a GMO, but it has limited host range, relies on food grade conjugative plasmids, risks of unintended gene transfer. In the case of plasmid based homologous recombination, we can have targeted modification. This is widely used in lab. Conditionally replicating vectors improve efficiency. However, it has low efficiency and it is very time consuming. We have high false positive rates and it is considered as a GMO. In fact, recombinering and CRISPR-9 engineering also suffers from the same GMO tag. So, in recombinering we can get targeted modification. It avoids cloning of homologous regions suitable for high efficiency and genome editing, but it requires recombinase optimization, limited plasmid stability and considered as a GMO. And CRISPR-Cas9 engineering, it is a versatile technique for gene deletion, insertion and silencing, high precision and efficiency, no genome scars is seen. Risks of off-target mutations requires replicable plasmids considered as a GMO, limited applications across the lab strains. Now, let us look into the potential applications of genetically modified lab in dairy fermentation. For example, if we look them from the perspective of various traits and applications and some of the examples of these strains, when it comes to safety, the application of these lab, particularly Lactococcus lactis and L. plantarum, is antimicrobial peptide production and degradation of harmful substances. And in the case of quality, we have lower post fermentation acidification like L. plantarum, then degradation of indigestible polysaccharides like L. rhamnosus, and from a sensory point of view, flavor aroma improvement by autolysis and production of aroma compounds possible in the case of Lactococcus lactis and Helveticus, then degradation of undesirable flavors like L. casei, then enhanced sweetness like Streptococcus thermophiles, then improved texture L. casei and L. Helveticus. Then we have these stress tolerance, particularly growth tolerance to stress like temperature, oxygen, pH, bile. We have L. gasseri, L. acidophilus and L. casei. Then we have certain valuable products like the production of micronutrients, sweeteners and chemicals by L. reuteri, L. lactis and L. helveticus. Let us now discuss briefly about the exploration of synthetic biology in food fermentation, which can help us in engineering the microbes. Synthetic biology enables modifying microbes to enhance fermentation by expressing genes that produce specific flavors, example heme for meaty tastes, and nutrients, example polyunsaturated fatty acids for health benefits. It can also help us in improving the nutritional value. Engineered microbes can address nutritional deficiencies by producing essential fatty acids and other viral nutrients. It can help us in enhancing yield and quality both. Optimizing metabolic pathways in microbial cell factories increases the yield and quality of fermented products, improving consumer appeal. And it can help us in achieving sustainability, since synthetic biology reduces reliance on traditional agriculture, lowering land use, greenhouse gas emissions and environmental impact while meeting food demands. So, some of the emerging trends and innovations of engineered food fermentation are the development of customized strains, genetic modification and selective breeding enhanced fermentation by improving flavor, nutrition and probiotic benefits. Then the optimization of control conditions, like advanced technology enables precise control over fermentation, ensuring consistent high quality products like sourdough with specific taste and texture. It can also help us in getting novel ingredients using alternative substrates such as brewing byproducts reduces waste and creates unique flavors like spent grain sourdough, and then it can help us in achieving health innovations. For example, engineered fermented food now targets specific health issues such as gut health or immunity with enriched probiotic strains, and it can help us in achieving sustainability. Fermentation focuses on sustainability by using waste materials and optimizing processes, reducing environmental impact and fostering innovations. Let us now go to section 2, where we discuss about the precision fermentation. We will discuss about the technological approaches for precision fermentation. First, let us begin with the definition and overview of precision fermentation. Precision fermentation involves optimized fermentation processes using specially designed microbial hosts as cell factories to produce high value functional food ingredients with high yields and purity. Basically, it utilizes microbial metabolism for the chemical transformation of organic matter, and the aim is to create products with enhanced health benefits and improved organoleptic profiles. It involves techniques such as high throughput screening and CRISPR-Cas9 for strain engineering. Some of the applications include precision fermentation, supports the sustainable production of enzymes, lipids, vitamins and flavoring agents often more efficiently than existing traditional methods. So, now if we make a comparison between the traditional fermentation and precision fermentation, and we look into the aspect of process control, traditional fermentation is often artisanal, spontaneous and undefined. It relies on natural microbial populations and backsloping techniques. Precision fermentation employs optimized methods including high throughput screening and genetic engineering to control and enhance fermentation process. With regards to product consistency, traditional fermentation can lead to variability in product quality and flavor due to uncontrolled fermentation conditions. Precision fermentation aims for consistent and improved product yield, quality and safety through precise microbial manipulation. And with regards to technological integration, traditional fermentation typically lacks advanced technologies and relies on historical cultural practices. Precision fermentation integrates modern techniques like CRISPR-Cas9 and multi-omics to enhance fermentation efficiency and product characteristics. So, the various techniques and technologies, for example, here we can have a overview of the use of precision methods on fungal fermentation, which uses CRISPR-Cas9, high throughput screening, next generation sequencing, molecular cloning and multi-omics. So, some of the advantages are it increases the process efficiency, cut down the cost, it increases yields, enhanced safety, the nutritional value is also enhanced, the organoleptic profile is also enhanced. Now, the CRISPR and genetic engineering for precision fermentation are essential for precision fermentation, enabling high-end accuracy modifications of microbial genomes to develop strains that efficiently produce desired food ingredients. Then we have the strain engineering, which involves selecting and modifying specific microorganisms such as grass, those are generally recognized as safe, bacteria and yeast both, to enhance their production capabilities under optimal fermentation conditions. Then we have the application in these microbial strains. Engineered strains can produce alternative proteins like those from Pichia pastoris used to create meat analogs, showcasing how precision fermentation can offer sustainable food sources while mimicking traditional ingredients. The CRISPR-Cas9 tool has been utilized to create yeast strains that express recombinant enzymes from other organisms such as Rhizopus oryzae to enhance sour beer production with unique flavor profiles. So, this table shows conventional fermented food beverages prepared using precision methods. In the case of bread dough, while Saccharomyces cerevisiae strain from a traditional Korean microbial fermentation starter Nuruk was used, and CRISPR-Cas9 genome editing was deployed, disrupting the RGT2 and SNF3 genes, which elevated glucose repression, resulting in increased carbon dioxide production during bread dough fermentation and improved strain leavening compared to the parental strain. Then we have the wheat based, bread and potato based chips, wild Saccharomyces cerevisiae strain from a traditional Korean microbial fermentation starter, similar to the earlier one. Here again we used CRISPR-Cas9 genome editing for overexpressing ASP3 and deleting URE2 or GZF3 significantly for increased asparagine consumption, leading to reduced acrylamide content in the baked bread. And in the case of wine, Saccharomyces cerevisiae Lalvin strain EC 1118 was genetically engineered using CRISPR-Cas9 editing technique for disrupting the ECM33 gene in these strain, which improved fermentation efficiency by 20% in nitrogen limited and 13% in nitrogen sufficient media under continuous shaking. The mutated yeast also settled faster, resulting in clearer fermentation media and larger yeast sediments. Let us now discuss about the importance of fermentation monitoring and control, where we use sensors, which are basically electronic noses and tongues. These multi-sensor systems are effective for monitoring fermentation processes, allowing for the detection of volatile organic compounds that indicate fermentation quality. And we get rapid responses. The use of these sensors enables for quick identification of deviations in fermentation conditions, which is essential for maintaining product quality and preventing resource waste. And the automation is largely beneficial. Integrating automation with sensor technology facilitates precise control over fermentation parameters, reducing the need for manual intervention and minimizing downtime. Real-time expert systems like GenSims 02 are increasingly being applied to fermentation processes, as these systems integrate data from advanced sensors, utilize improved signal analysis and control algorithms, and apply artificial intelligence techniques. This approach enhances our understanding of fermentation behavior and enables more precise monitoring and control of bioreactors. Let us discuss about the applications in functional food, production of functional ingredients, particularly bioactive peptides, vitamins, antioxidants, and others. So, in the case of bioactive peptides, engineered Saccharomyces cerevisiae produces bioactive peptides that boost immune function and lower blood pressure. Then Aspergillus oryzae is used in precision fermentation to produce essential vitamins like riboflavin B2 for functional foods. Then in the case of Xanthophyllomyces dendroros, it generates astaxanthin, a potent antioxidant for incorporation into health focused food products. Then besides this, precision fermentation also produces enzymes and compounds that enhance food preservation and nutrition. So, tailoring fermented food for specific health benefits today is a reality. It helps us in enhanced nutrition and targeted health benefits, then flavor improvement, and also long-term sustainability. Precision fermentation engineers microbial strains to produce nutrients like vitamins and bioactive peptides, as we have discussed in the earlier slides, which boost the health benefits of fermented foods. Modified yeast and fungi can target specific benefits such as probiotics for gut health or bioactive compounds for immune support. And CRISPR-Cas9 techniques enables the development of strains that enhance both nutritional content and flavor, making functional foods more appealing. Precision fermentation promotes sustainable food production by reducing reliance on traditional agriculture, and all of these in brief provides us healthier, tastier, and more sustainable dietary options. So, now there are many companies which are involved in fermentation or production of food ingredients. In the table you can see companies like Arjada, Peprotech, Richcore Life Sciences, Remilk, and so on, and also they are classified into companies which some of them produce strains and recombinant proteins, some of them produce only proteins, some of them are focused on production of lipids, and companies like DuPont also produces probiotics for food, beverages, and dietary supplements. So, for example, Arjeda goes for designer fermentation strains for ingredients. Then you have the Peprotec, which is focused on recombinant proteins including growth factors for cultivated meat, and similarly Richcore Life Sciences also has similar focus areas. Then you have Biosynthia, which uses mycelium as platform for animal muscle proteins in meat alternatives. Then you have Remilk, where dairy proteins through microbial fermentation is being done. And then you have Final Foods, whey proteins produced by yeast for CEs. Then Better Dairy produces dairy proteins using precision fermentation, and Fumi Ingredients trying for egg replacement ingredient produced by microbial fermentation. Then we have companies like Melt and Marble that focuses on recreating animal fats using precision fermentation, and Sense Foods are trying to produce fats by fermentation for dairy products, and Yeli Bio focuses on precision fermentation derived fats for animal free products. So, this table looks like very imaginary, but many of these are being now possible. So, in future we may be able to reduce our dependence on limited resources like land and water if many of these things become scalable and also accepted by the consumers. So, monitoring and artificial intelligence application in fermentation is one of the recent advances. Here techniques like high performance liquid chromatography and infrared spectroscopy improve process control by accurately measuring products and substrate concentration. AI is getting integrated. Machine learning methods such as XGBoost and random forest are used to predict optimal fermentation conditions that can be analyzed in Periscope Opt. Periscope Opt is a machine learning based platform for predicting optimal fermentation conditions and yields for recombinant periplasmic protein expressed in Escherichia coli, enhancing efficiency and scalability in fermentation technology. So, what are the key considerations for introducing precision fermentation derived ingredients into the market? As I told in one of the slides earlier, that in future probably we can reduce our dependency on limited resources like land and water to a large extent by producing many of the food ingredients through precision fermentation, but this has to be actually accepted by the consumer finally. So, some of the important considerations apart from these is the policy. Number one, for example, we need to think about the safety, environmental and ecological risks, then regulatory control is very essential, labeling is very important, and the consumer perception. Then we need to look for market opportunity, whether really it exists over there, and then because these are very advanced kind of biomanufacturing, we would require a lot of investment and also companies which need to come forward. And in the earlier list table you have seen actually there are so many different companies focusing on different areas, like some focusing on proteins, some on lipids, and others on other bioactive compounds. So, more companies in future probably would be required. And then the IP landscape scenario is very important, because each and every of these product processes and even those strains which are developed by different methods need to be protected by IP, and the know-how also is an important aspect, because the know-how is never mentioned either in the scientific papers or even in the patents. So, finally, of course, the fermentation will be carried out by the microbes. So, strain selection in engineering, the choice of feedstock and the fermentation process, and finally downstream processes which will give us the final product, and scaling up of the entire processes is going to be a very, very important consideration. And then its application in food, particularly the ingredient specifics or specification, the physical functionality, and then nutritional properties, safety, whether it is fit for consumption or not, is going to play a very, very critical role, particularly with respect to precision fermentation derived food ingredients. So, with these we come to end of today's lecture. Thank you for your kind attention.